Good morning, everyone, and it's a privilege and a pleasure to present at the Macular Disease Society conference today. And so I'm Andrew Webster. I work at Moorfields Eye Hospital and one of a team of 5 consultants concerned with genetic eye disease, one of the largest in the country. So we see many people with genetic disorders, including macular dystrophies. I also work at UCL, at the Institute of Ophthalmology, which is a unit next door concerned with research. So during this talk, I've been asked to talk on knowing your genes. I'm going to keep it as simple as I can. There are very few slides. I'm sensitive to the fact that a number of folks won't be able to see the slides. So there's nothing important on the slides, merely bullet points to help me keep my talk organized. But during the talk, I want to cover the following things. Firstly, what are genes? So I'm talking about knowing your genes. I need really to say a bit about what genes are. And I apologize to anyone with an MSc in molecular genetics. It will seem patronizing, but I want to try and just give an overview to the layperson of what a gene is and why it matters. So why are genes important in the disorders that affect people who are members of the Macular Society? How does one find a gene? Now, there are 2 questions there. How did scientists first find the genes that cause eye diseases, such as macular dystrophy? And secondly, how does a person find their own gene when they have such a disorder? So I want to cover those 2 questions. And why is this a thing worth doing? So it's quite a big job to try and find the gene causing a person or a family's condition. And what's the advantage of that? And I just want to stress what we can and can't do with that information and why it might help someone. So firstly, what are genes? This is the difficult bit. I'm going to assume people don't know anything about genetics. So all cells in all organisms and all living things, and that includes viruses, have genes. It's what defines life. And gene or genetic material is made of the same stuff in all of those organisms and viruses. And that, and people will know this bit, is called DNA, deoxyribose nucleic acid. Very rarely, organisms have RNA, which is a related molecule comprising their genome. Some viruses, for instance, are RNA viruses. But in all organisms, there is a polynucleotide sequence that comprises the genes of the organism. And that DNA, or indeed RNA, is made of 4 components, 4 different components strung together in a long linear sequence. And those components are called adenine, cytosine, guanosine, and thymine. Now we abbreviate that to AC, G, and T. So these are molecules. Everything's composed of atoms and molecules, including humans and their DNA sequence. And they're specifically different building blocks that make up the DNA. And they comprise a linear structure. It has direction. It goes in one direction, and we need to know that as geneticists, which direction we're talking about. So the precise order of that sequence matters and determines what the organism is. DNA is very often split into chromosomes. People will have heard about chromosomes. And in humans, we have 23 pairs of chromosomes. A bacterium would have just 1 single chromosome. A fly has 4, a rice plant has 12, and I found out this morning that a pigeon has 40. So when we look at the humble pigeon, just remember that the pigeon has more chromosomes than we do. Now, in all of those organisms, except bacteria, they're paired. So chromosomes exist in pairs. So each cell has 2 copies of the genetic sequence, 1 from each parent. And so we have 23 pairs of chromosomes. So in all, each cell has 46 chromosomes. And all of this material is contained within a small part of each cell called a nucleus. That's what the nucleus does. And the nucleus is only 100th in diameter, about 100th of a millimeter large. So packed within that small part of a cell is all the information to make the organism and all the information that makes you different to the person next to you. So why are genes important? So genes determine most things about us. And if we look at monozygotic twins, that makes us realize just how powerful genes are in determining traits, for instance. So height, eye color, hair color, stature, facial appearance are determined to a large degree by the genes that the person inherits from their parents. In medicine, disorders can be divided into two types, common or complex disorders or rare disorders. And complex disorders are those such as rheumatoid arthritis or inflammatory bowel disease, type 1 diabetes, type 2 diabetes. The majority of disorders which doctors and medicine are concerned about are complex diseases. Even infections have a genetic component, so the immune system is very complex, and that will determine to what degree a person is susceptible, for instance, to bacterial and viral infection. Now, these common disorders are due to genes, but they're due to many different genes. And of the disorders I've just mentioned, one that is most understood, arguably, amongst them is age-related macular disease. And so that disorder is due to genes, and we know a lot about the specific genes that cause it. And that has been worked on since 2005, when those genes were first discovered. And that helps us greatly in understanding the disorder. And more recently, it's helped develop trials to try and find better treatments for it. It could, in theory, allow us to tell what the risk is of a person getting age-related macular disease. That's not something we do because we can't do anything active about it, but theoretically it's possible. We know enough about the genes that cause AMD to give a reasonable risk of a person's susceptibility to the disorder. But here we're concerned with rare disorders or Mendelian disorders, after Gregor Mendel, who first described the laws of genetics many, many years ago. And so these disorders are due to just one gene. So of all the genes in our genome, in rare disorders, it's one gene that is responsible. And that gives us an opportunity because we know exactly where to aim any novel treatments at. It's like having a car that's not working. If it's just old and decrepit, with many things going wrong with it, it's hard to fix. But if it's just one component that's stopping the car working, and we identify the component, we can aim our remedy to it. So from now on, I'm going to be concerned with those disorders, and many people on this call, not all, but many will be affected by one of the conditions. Now, we have 20,000 genes in our genome, and so we need to know, if possible, when we see a person that we think has one of these rare genetic disorders, which of those 20,000 genes causes the condition. And we know where to look because we know many of the genes now that cause inherited eye problems, including macular dystrophy and cone dystrophy and RP, retinitis pigmentosa. And those genes can behave in two main ways. They can be dominant or recessive. And what that means is that in our pair of chromosomes, a dominant disorder will occur when just one of the pair has the change in the genetic sequence in that gene. And examples of that are macular diseases such as Best disease, Salisbury disease, dominant drusen. And the most common, which doesn't have a easy to remember name, is RDS peripherin 2. Many people have that condition. That can be inherited through families. An alternative way of having a problem is recessive disease, in which you need not just one, but two changes, one in each of the pair of chromosomes and in each of the pair of genes that you inherit from the parent. And ABCA4, which is the most common gene that causes macular disease, that causes Stargardt disease, macular dystrophy, cone dystrophy. It can be given different names, but now we're tending to label it as ABCA4. That is such a gene. You need a mutation, a change in the gene in both mother and father to have the disorder. Now this is, I've just changed slides now, and the first time there's actually a picture on it. So I just want to talk a bit about how we find a gene and how complex that is. And on the slide, there is a picture that I've downloaded from Google Maps, a journey that I could take from Moorfields in London to Moscow. I've chosen that because it's 3,000 kilometers, so it's very easy to use this analogy. Because we have 3,000 million letters in our DNA code, and so we can use this to try and just understand the vastness of our genome in terms of information and in terms of finding out what might cause a genetic disorder. Now, I'm old enough to remember the olden days before the Human Genome Project completed. It first completed its sequence in 2003, but thereafter updated a number of times. But before then, the challenge was to find a gene for a disorder. And so we'd have to look at different genes and try and find the spelling mistake in affected patients to confirm whether or not that candidate gene caused the condition. And so to give you an idea of the scope of that, if I was to set off on a journey between Moorfields and Moscow, there are 20,000 genes, so we would come across a new gene every 15 kilometers of our journey. But the size of the gene is much smaller than that. The majority of our DNA sequence doesn't actually encode for genes, it encodes for intergenic DNA. Much of that DNA doesn't actually do anything, but some of it is important in controlling how those genes are expressed. Now remember, all of our cells have the same genetic code in them. That doesn't vary from nucleus to nucleus, it's the same. So something has to determine what makes a bone cell different to a skin cell, different to a photoreceptor cell. And that difference is due to which of those 20,000 genes are turned into RNA and protein and actually function within the cell. And the sequences that determine that are those sequences between the genes, so-called promoter sequences. So a lot of that intergenic DNA sequence is important, but much of it isn't. So every 15 kilometers we'll come across a new gene. But the gene itself is smaller than that. So for instance, the ABCA4 gene, which I've mentioned is the most common cause of macular dystrophy, Stargardt disease, would be 128 meters long. So I'd start my journey from this very office, and by the time I got to Wall Street Station, I would have completed the distance of the ABCA4 gene. If it were there, of course, it could be somewhere in Poland. Alternatively, the gene that causes dominant macular dystrophy, PRPH2 RDS, is a lot smaller. ABCA4 is a large gene, many genes are smaller, and that's only 25 meters long on this journey. So we'll have completed that distance by the time I get to the lift from my office to get to the ground floor. But the most important statistic to remember is that one letter, one unit of DNA on this scale, which determines whether a person has a disorder or not and would need to be scrutinized on this long journey, is a millimeter in size. So if someone has ABCA4, they'd have 2 of those going on in that journey in the ABCA4 gene. If it were a dominant disorder, there'd just be 1. So we'd need to scrutinize, theoretically at least, every millimeter of our journey to find out which particular mutation and thereby which gene causes the disorder. So how does one do that? It's now a lot easier than I suggested with that analogy. So we now know the human genome, and that was first completed in 2003. I remember when that was first announced. A very rough sequence of the human genome. The Human Genome Project started in 1990 and comprised many countries and laboratories, and it cost many millions of dollars to complete. We now know the genes that cause the majority of single gene disorders in humans. So there may be a few we're yet to find, but we know most of them. So that makes our task easier. So we can already identify the subset of genes that might cause a person's disorder. And in the UK, we have specific panels of genes that we can apply to the human sequence of a person to constrict our search to them. Now, genetic testing in the UK has undergone a revolution recently to reflect the increasing technology. When I was doing this sort of work in the laboratory, we had to painstakingly make that very journey millimetre by millimetre. We used a technique called Sanger sequencing after Frederick Sanger, in which we would pick on a small bit of DNA to look at a separate time, perform a reaction, a PCR reaction and a sequencing reaction, and then read the sequence letter by letter. And we were limited in how much we could do in a single day. And so it was a very onerous task. Later, the revolution in the technology occurred, next generation sequencing, in which a machine could do that task in parallel. So many sequences all at the same time, registered on a computer, and we could train a computer, have a pipeline to look at that sequence and compare it to the reference sequence and highlight the changes in the whole genome now that might be causing the disorder. And then we can look at those changes and we can apply some rules as to whether or not any of them might explain a person's condition. So there is a reference sequence that's interesting in itself, and that changes. The more we get to know about the human genome, it's still evolving, then the reference sequence has to be updated. We're working with HG38, and previously we were working with HG19, and whenever we communicate genetic data, we have to be sure of which reference sequence we're using to denote the change that we're talking about. On the analogy, it's a bit like changing the border between Germany and Poland. It can cause problems and misunderstandings. Now, practically in England, there is now the Genomic Medicine Service split into 7 centres, and there's a similar structure in Wales and Scotland. So everyone with what is thought to be a Mendelian disorder, a disorder that's likely to be due to a single gene, can now get genetic testing. They can either be referred to a genetic centre such as Moorfields or others around the country, there are many, or else a clinical genetics service. And they can have, if they have a suspected retinal disorder due to a gene, they can have whole genome sequencing. So this is the new technique which looks at, with a few exceptions, looks at the whole genetic sequence. And then a computer pipeline is applied to that, and we get approximately over a 50% hit rate. It doesn't sound very high. It depends on what the condition is. It depends on a number of other factors. But we should get over 50%. That's higher than many other specialities. We know a bit more about the genes that cause eye disease than, say, the cardiologists will know about the genes that cause rare heart disorders, or nephrologists for kidney disorders. And that's increasing the more we do. And if we fail to find the change first time round with this computer pipeline, then there is the prospect of research being done later on a person's sequence to find the cause. And so people will be given the option as to whether or not to consent for that research. It's called the National Genomics Reference Library. And it's a safe thing to do. It's very well guarded within the Genomics England platform, but it does allow investigators such as myself and colleagues who know about this condition to look specifically more carefully at a person's genomic sequence to try and find the causative gene. Final slide now. So this talks about why we should know the gene. What's the point of knowing the gene? Now, the important thing is, and I'm sorry to give this news, but finding the gene doesn't make a condition treatable. Now, there's one exception to that at the moment, and that is a gene called RPE65. Some people will have heard of Luxturna, which is a gene replacement therapy, which is being done now within the NHS to treat those people with retinal dystrophy due to this gene RPE65. It's a recessive condition. It's a severe condition. It doesn't cause macular dystrophy. It causes a severe onset retinitis pigmentosa-like condition in which the dark vision is affected more than the light vision. The majority of people don't have that. We always check it, but the majority of people don't have that. And so at the moment, unfortunately, there aren't other treatments yet. So we can't make the disorder treatable. But there are trials ongoing. And many people will know that, and some people will have been enlisted to trials. Doesn't mean that they're treatments yet, and so very often a trial doesn't prove to be efficacious. But the more trials, the better. And so finding the gene allows people to, if they wish to, go forward to trials if they have a relevant condition. But it does help us make the diagnosis. And so doctors are always keen to make the diagnosis. We don't like doubt and ambiguity. And when talking to patients, it's a lot easier to do so and give information about the condition when we know what it is. And that completes a diagnostic odyssey, the so-called expression that's used a lot, in which people have a problem, but for many years don't know what causes it. And usually a person can deal with a condition when at least it's no longer a mystery. We know the gene, and we know that other people have it. We know to what degree animal models have been made, and whether there are trials or not going forward. We might also give some information about the likely future of the vision given the gene, but that's difficult because these things are variable. It helps us counsel the family. And so these disorders that we're talking about now are genetic disorders. And so a person will want to know what the risk is to their relatives, what's the risk to their child of inheriting the same disorder. Now we can make a good guess of that just by knowing about the family history, but to be sure, we would not need to know the causative gene. And then finally, research. So every time we do find a person's diagnosis in genetic conditions, we learn something. And so it's a cumulative endeavor. We learn more the more we find, the more patients we test. And so if the person is doing this, they're contributing to research. And we at Moorfields ask a person whether they'd like to be part of our research program in parallel. And that will help us go forwards with treatments in the future. So I've come to the end of my talk today. Thank you for listening. And the future Professor Webster will be available for questions. Thank you.